Good afternoon, everybody. This is Stefan van Sinvliet speaking from Biognosis. It's my pleasure to welcome you to today's webinar entitled Proteomics of Aging, Using Data-Independent Acquisition to Study Organ Aging. And it is my pleasure to have with us today Dr. Alessandro Ori and Dr. Joanna Kirkpatrick, who will present their work for our audience. Followed with a Q&A session where you can ask any questions you may have to Alessandro and Joanna. You can ask questions using the questions functionality on the right-hand side of your webinar interface. We are recording this webinar for you today, so if you have any questions, let us know at support@biognosis.ch. We will send you a recording, copy of the recording to all participants today. So with that, I'd like to hand over the floor to presentation mode to Alessandro and to Joanna, and I hope you will enjoy this webinar.
Okay, I hope that you can see my screen. And so my name is Alessandro Ori, and I am a junior group leader at the Leibniz Institute on Aging. And I must admit this is my first webinar, so it feels a bit weird to talk to my computer, but I hope that you will enjoy the next 30 minutes. So this is a bit of an outline of what we want to show to you. So I will have at the beginning a little bit of an introduction on aging research and how we are planning to use proteomics technology to study organ aging. In the second part of the talk, we are going to give you a little bit of details about our experience in setting up data-dependent acquisition in our lab. And for this part, I will hand over to Joanna, who is sitting here next to me, and she is the manager of the Proteomic core facility here at our institute. And in the final part of my talk, I'm going to show you how we use data-dependent acquisition to study aging of the intestinal epithelium. So, as I said, a little bit of introduction about aging research in general. And so this is one of the major topics of our institute, and this is mainly driven by the dramatic demographic changes that particularly Western countries have been facing in the last 100 years. And this is what is shown here in this graph, which is showing essentially a reversal of this period. So at the bottom you have young ages, at the top you have old ages, and as you can see, at the beginning of the century, majority of people were accumulating in this young age, while the projected statistic for 2016 see a reversal of this pyramid, with many people in the old age being the most represented age group on our planet. And this, of course, has implication also for our healthcare system, and this shows what is shown in this second panel here, which essentially shows a projection, again for Germany, that shows essentially an increase of the age group of patients that are hospitalized, meaning that by 2013 we're going to have more and more elderly people in our hospital, and this of course is going to have massive implication for the cost of our healthcare system. And this is why aging research, we believe it's important. It's important to also perform research, basic research, as we do, in order to essentially being able, perhaps in the future, to develop therapies that improve health at old age, because this is going to be the majority of the patient that will need to be treated in the future. And on a little bit more mechanistic point of view, what is the main problem? And the problem is that while we age, there is an inevitable decrease in the function of almost all our organs. So this is shown in this plot on the left-hand side here, where you have age and you have an estimate of the maximum capacity of an organ function. And as you can see, and probably you can also experience during your life, it doesn't matter what kind of organ you look at, this is an inevitable decrease in organ function that occurred during age. And this goes hand in hand with another major problem, which is the increased risk of disease. And this plot on the right here shows, again, having age on the x-axis, shows the increase of incidence of risk of incidence of cancer in the German population that dramatically increase after the age of 50, inevitably for both male and females. And so the core of our research is trying to understand what is happening at the molecular level behind this decrease in organ function. And what is also becoming very clear and established in the field of aging research is that stem cell, and here I'm talking about adult stem cells, which reside in the majority of our organs, plays an important role in the decline of organ function. Meaning that it has been shown in many organs that a decline in stem cell function occur during aging. And this is particularly relevant for organs which have a very, very high turnover, such as, for example, the intestinal epithelium, which is something where our research focuses on. And why we decided to focus on this organ? And one of the major reasons is that the intestinal epithelium is one of the most important interfaces of our body. First of all, it's not only the site of food absorption, but it's also an important interface with the microbiomes, with the bacteria that populate and colonize our intestinal tract. And of course, these two functions are inevitably linked to also other organ function within the organism. So, meaning that the health of our intestinal system is necessarily linked to the health of the whole organism. There are also a couple of other interesting points, one of which is that the intestinal epithelium is an extremely dynamic organ. So if we would open it flat, the surface area would be equivalent to pretty much the size of a tennis court. And what is very interesting is that in human, this tennis court in our gut is renewed every 3 to 5 days. So the entire epithelium surface is renewed every 3 to 5 days. And this all relies on the function and controlled proliferation of intestinal stem cells. So I want to give you a little, one more introductory slide to show you a little bit of the anatomy of the intestinal epithelium. So this is a cross-section across the small intestine epithelium. And this is an electron microscopy picture that shows on top the villi, which are responsible for forming this amazing surface area, which I mentioned to you before, which are covered, you can see this cartoon here on the right, by enterocytes. And underneath this villi, there are these little structures here, which are called the intestinal crypts, and that is where essentially where the intestinal stem cell resides. So these intestinal crypts form the so-called stem cell niche, where intestinal stem cell proliferation occur. And intestinal stem cells are, of course, able to differentiate in all the different cell types that form the epitheliums, and they are pushed toward, higher toward the villus to form, to cover the whole epithelium. And during the last year, there have been evidences that many interventions, such as, for example, changes of diet, can influence the function of the intestinal stem cells by either modulating the stem cell activity itself or by affecting the function of neighboring cells, such as the spined cells, that form the niche for the intestinal stem cells. And of course, I wouldn't be talking about this if it wouldn't be the case that there is a loss of intestinal epithelial homeostasis during aging. And this has all to do to the delicate balance in the control of the intestinal stem cell, that on one hand could lead, if not regulated enough, to hyperplasia and predisposition to cancer, and on the other hand, if this regeneration capacity is reduced too much, can lead to food malabsorption and loss of barrier function, leading to, for example, chronic infections in elderly people. And the aim of our work is to identify proteome alteration that have a causative role in this decline of organ function, and ideally to identify reversible alterations that could be intervention points for the development of future therapies. And the way that we want to do it is to use the mouse intestine as a model system, and it's essentially very simple. So we want to take organs from young, healthy individuals and compare it to the one of old individuals, in order to try to understand what makes this old organ more prone to disease as compared to the young. And as I mentioned, we want to use it to do it using proteomic technologies and then applying bioinformatics tool that would allow us to identify essentially molecular networks that are affected during the course of aging. And a little bit more in detail, I mentioned before that the stem cell can be influenced by either intrinsic factor in the stem cell, but also by their environment. This is why we decided to analyze not only the stem cells by themselves, that can be isolated from the organ, but also the intestinal crypt, which I remind you for the niche for the stem cell, as well as the whole organ, because we believe it is important, and proteomics give us this opportunity to identify both changes that occur within a cell, but also changes that occur outside the cell in the extracellular matrix. And what we want to compare many different samples. We want to compare samples from animals of different ages, but also compare the effect of different diets and perhaps study what makes certain mouse genotypes more or less prone to develop disease. And as I already mentioned, I already spoke about multiple sample type and multiple different comparisons that we want to do. And this makes actually an excellent motivation to employ data-independent acquisition as our primary choice of technology to compare the proteome profiles of these different samples. So now that this introductory part is over, I would hand over to Joanna, which is going to give you a little bit of details about our experience of setting up data-independent acquisition in our lab.
Okay, hi everyone. Yeah, so this will be a little bit more technical and less biological, but just to give you a basic overview of how we've started and how we've moved along with our workflow. So, yeah, like Alessandro mentioned, we are going to have a lot of samples and they're going to be coming to us over potentially a wide time frame. So then it made it much more suitable to this DIA workflow rather than trying to collect everything together and waiting for a label type TMT experiment, for example, where we have to pool all the samples together at the same time. This way we can take representative samples across the different types, so we can have the dietary restricted ones, the different age groups, and first of all we can create libraries of spectra from all of the different sample types, so that we have a library in the end that contains peptide identification information from potentially any type of sample for which we might then further wish to analyze. So old samples may contain different proteins to the young samples and so on and so forth, but we have a library containing a representative data set from all of those. And in the first instance, one has to still perform a data-dependent acquisition in order to generate these libraries, so a standard MSMS experiment to build the library. But once we have that, then we can then take individual samples and acquire data in the data-independent acquisition mode, match this data using the Spectronaut software to the library that we've created, and it uses all sorts of features from the data, the mass, the charge, the retention time profiles, to allow us to do both identification and quantification of the peptides, and therefore the proteins within our samples. And so we can keep adding to the library if we want, but it also allows us that we can have a stock library, and as samples come in over time, when experiments are finished, we can just keep analyzing new data-independent acquisition modes and referring back to this library that we've created. So if we move on to the getting more technical now. So this is something I put together when I was thinking about how one starts off in the beginning and what you have to do every time and what you only have to, let's say, do once for your system in your lab. So with each sample, there's a little code down the bottom. So the green arrow boxes are something that you obviously have to do with every sample, so the standard protein extraction and digestion. But then there are other things that one only has to do once in order to set up your system in your lab. So one important factor is to calculate the dead time on your HPLC system that runs in front of your mass spectrometer, and to optimize your gradients and your MS2 DIA segments for the best performance that you can get out of your particular machine in your lab. These are something that you can do on a representative sample that you want to then further analyze, and once you've done it, you don't need to keep repeating this. When you want to create a library or you want to use a sample to add to your library, then you'll always have to run a DDA run under these optimized conditions for adding it to the library, and obviously search that data to allow it to be included and then put into Spectronaut. And of course, for every sample, one has to spike in the HRM kit peptides from Biognosis into the sample and acquire the DIA data. And we always do this in technical duplicate, and we find that this actually improves the robustness of our statistics in the end. And of course, yeah, one has to look at one's data in Spectronaut at the end for looking at the identification and the quantification of the proteins in each of your samples. So we're lucky that the guys at Biognosis have done a lot of work in already providing us with a good starting point for generating a DIA method. And the sort of fundamental factors which one needs to consider when you're setting it up is that the performance of what you get out in the end will depend on majorly on these four criteria. So the number of windows of DIA that one chooses to measure, the resolution on your scan, so how high you have the resolution in your Orbitrap in our case, how long you choose to run your LC gradient for, and how you sample your chromatographic peaks within that time. So for us, it's obviously critical that you check your chromatography so you know the dead time of your system and also the peak widths. MS1 and MS2 resolution should be optimized for your instrument, and we spent a lot of time in the beginning playing around with the best compromise between obviously speed of acquisition and then accuracy of the measurements and how well our libraries perform in the end. And obviously, based on your chromatography, the cycle time that gives you enough data points per peak to do reliable quantification. And it depends on your instrument, but there are some settings that one might have to do manually on some instruments that are filled in by default on others, depending on what you are working with. So just for your information, we are working on a Waters Nano Acuity UPLC in front of the Thermofusion Lumos. So we have a number of, let's say, additional possibilities that we could consider. So, but we know things about our chromatography. So we have a 7-second full width at half maximum for our 2-hour gradient runs. We found optimally that MS1 resolution in the Orbitrap of 120,000 and MS2 resolution of 30,000 worked best for us. This is, just so you know, always done with HCD fragmentation, and because that won't be an option when you have your Q-Exacta, for example, but of course one can choose between CID and HCD on the Lumos, but we found that HCD definitely works best. To get our data points, we were working with a 2-second cycle time, and we calculated that would give us somewhere between 20 and 25 DIA segments. And we tried both in the beginning to see what worked best. We thought 25 would actually be overkill and we'd have to go somewhere in between, but in the end it worked out fine for us, and that leaves us with about 6 to 7 data points per peak, which is sufficient. On the Lumos, for anyone else working with the instrument and the, let's say, original version of the tune, some of the things in the methods you have to set manually, and we had to set our fill times manually for the MS1 and MS2 in order to allow us to fulfill these cycle time requirements, and that was set to 80 milliseconds. And it's also important that you manually set the default charge date to 4, and this was on the advice of the guys from Diagnosis, but it's not so easy to find in one's Lumos. So just for those people who are editing methods, I took a couple of screenshots. So on the left-hand side, this is where you find it in the global parameters, and I guess most people are only looking in their methods at the scan parameters, which is the next tab across. And then one has to scroll down to the MS little globe here on the left-hand side, and this is where one finds one's default charge date setting. So it's important to set that to 4 for these types of analysis. Normally it's default set to 2 when you make a new method. So that's just for the geeks amongst us. And then this is how one's MS1 level scan parameters look on our instrument. So again, here's where you can set the Orbitrap resolution for your MS1. The mass range here also we set by hand. We have also had to set the injection time by hand. So these are just things that with the current version of the tune on the Lumos, one has to do quite a lot of manual entering of information here because a lot of the defaults are not set as standard. And then secondly, we move on to one of our DIA segments here. So you see our precursor mass range. We have an isolation window of 60 m/z on this particular segment. The default when you start a DIA method on the Lumos is that it creates UCID, so you have to switch this to HCD. You also have to set the scan range manually. This is not done automatically, and again, one has to manually input one's injection time. I'm reliably informed this will change with the latest versions of the tune, but we wait and see. Okay, so just to kind of summarize and give you some tips. When you're making your library, we discovered it's important by process of not having done it the first time to have the HRM kit peptide spiked into your sample at this point. It's useful for Spectronaut to be able to recognize them. Then it's also, if you're running a Lumos or a Fusion and you have the option, it's better to run in Orbitrap mode rather than Orbitrap ion trap mode, and also to select HCD fragmentation. These will both be defaults on a QE, for example. But it will mean that your spectral library will be better suited for the type of DIA data that you're going to acquire. The recommendations are to inject around 1 microgram per column, but it's worth checking your system and the best loading performance for whether this makes sense for you. So some people talk about more. We inject routinely slightly less for our best performance. And in the beginning, we just took our best performing MSMS method that we had with HCD fragmentation and MS2 measurement in the Orbitrap. And we just edited this slightly by increasing the HEC settings for the MS2 compared to our normal settings, as we found this really helped us in our library creation and then also in our DIA experiments. It is optional when you're creating your library to pool some of your representative samples and then fractionate. We would do this by high pH offline fractionation and acquire a library on the fractions. Or you can just run a representative number of your samples directly in DDA to create your library. And with the future data that we will show you, this is exactly what we have done. Then, of course, one searches one's data either in MaxQuant or PD. So we use MaxQuant, and you use the results to directly import a library into Spectronaut from the output. This is very intuitive and straightforward in the software. And other things that we've learned is that it's important when you do your search to take the FASTA file of the HRM kit peptides and include them in the search so that the software recognizes that they're present in the data files right from the beginning. I learned by having deleted my raw data because I ran out of some disk space that if you are importing your MaxQuant output into Spectronaut, you should just leave the raw data there until you finish making your library. Otherwise, it tends to get a little bit upset that it can't see the raw files. Once you have your library, you can then remove the raw data These are just things that we have learned from experience can help hopefully avoid other people making the same mistakes in the beginning that we made. So then I will hand back to Alessandro shortly, but this is just a summary of what we have. So the best conditions for DIA in our hands on these crypt samples were 2-hour gradients, although we tried different times, the 120,000 resolution in the MS1 and the 30,000 resolution in the MS2, 25 segments in the DIA, and 80 millisecond fill times. And then just to give you a summary of how our library looks under these conditions and how the recovery of the library looks with some of the samples that we've already measured. So we included in our library 22 representative samples from across age groups and also dietary conditions. We used directly to create our library, so without any fractionation. And in this case, our library now consists of more than 98,000 precursors, which correspond to over 70,000 peptides from nearly 6,000 proteins. And the library recovery from DIA measurements, which are carried out in technical duplicate, is around 75% on each sample. Back to Alessandro.
Okay, cool. Thanks a lot, Joanna. And so now, in the next slides, I want to show you a little bit how we used this setup that we did to analyze the intestinal crypt proteome. So we did quite some effort to set up this method, and we wanted to have a little bit of a proof of concept to see how indeed acquiring these DIA runs across many samples could work. So we started with a small pilot experiment where we took the small intestine from a mouse and we divided in 12 sections. Each one of these was around 1 centimeter. And we isolated intestinal crypts for each one of these sections, prepped the sample for mass spec, and analyzed each one of these sections in technical duplicate using our library that we generated. And so in this next slide, what I'm showing you is a heat map, which is generating by calculating Pearson correlation coefficient across any two of the single run that we analyzed. And the runs are labeled according to the section numbers. So section 1 is the most proximal to the stomach of the small intestine, and section 12 is the most distal. And as I mentioned to you, for each sample we measured 2 technical replicates. So we had in total around 5,700 protein groups that we cross-quantified across these 24 samples. And the heat map, you can see that the green color corresponds to higher value of Pearson correlation. And you can see that we have an excellent correlation between technical replicates. As you can notice that they always cluster together with the extremely high value of Pearson correlation. And I must mention that we scrambled the acquisition, so there is not the technical replicates were acquired as successive injection, and this I think shows the potential of the measurements. But getting a little bit more into the biology, so you have on the right hand side here, this is the schematic of more or less the 3 major anatomical regions of the small intestine. And as you can see that just by looking at the correlation between samples, we can retrieve these anatomical differences that are highlighted by this color on the heat map. We have a little bit of an outlier sample here, which is the one that is most proximal to the stomach, but this is because we believe it is indeed a little bit of a different sample as compared to the other. So meaning that protein profiling using DIA allow us to recapitulate anatomical differences in the intestinal crypt proteome. And of course, once we have such a large matrix, what an example of what we can do, and this is what I can show in the next slide. We can run algorithm, for example, Kmeans clustering, to identify proteins that show similar expression profile. And in particular, this cluster here is a cluster of protein that shows, let's say, a gradient of increased abundance going from the duodenum to the ileum, toward the ileum. And in this big plot here, I'm showing one of such example, where each one of the bar here, there are 2 bars for each sample due only to the technical replicate, and on the y-axis you have the protein quantity as directly exported from spectrum now. And as you can see, this protein shows a nice gradient across the tissue. And this protein was actually, I'm showing you here because it was actually often one of the few cases that was known to display such a gradient in the small intestine. But we have identified hundreds of proteins that shows this or other profiles across the small intestine. And interestingly, some of these happen to be actually extracellular matrix components. And this is telling to us that perhaps the environment of the intestinal stem cell might vary in the different region of the intestinal crypt protein. But I want finally to get to some aging data, and this is what I'm showing you here in this next slide. Again, using a very similar layout. So this is a heat map of Pearson correlation. And in this case, we analyzed intestinal crypts from 4 young animals around 10 weeks of age and 4 old animals of 26 months of age. In total, we cross-quantified again over 5,000 protein, and as you can see, the unsupervised hierarchical clustering can clearly separate young animal, 10 weeks here, again measured in technical replicate, from the old animal, which form a separate cluster, indicating that we can indeed retrieve the impact of aging on this protein profile. I also want to highlight a little detail. As you might notice here, the young animal form a very highly correlated cluster, indicated by the green color, while the old animals seems to form 2 separate clusters, and they don't have such a great correlation across each other. And this is something that we can also observe by looking at the distribution of the coefficient of variation for all the proteins across young and old animals. So in this box plot, I'm showing you the distribution of the coefficient of variation. You see that we are around 20% about proteins for proteins across young animals, and this significantly increased for all the proteins to around 30%, indicating somehow that there is more variability in the protein profiles from old animals than from the one of young animals. And this is in line with other evidence that people have observed before using transcriptomic that indicates that there is often more variability in the molecular profiles that one obtains from old animals as compared to the young one. And this might have implication related to the speed of aging that these animals are displaying. And of course, what was important for us was to be able to identify over 100 proteins which are differentially expressed in young and old animals, and this is what I'm showing in this volcano plot using the Q-value cutoff directly from Spectronaut. And these are, of course, for us very interesting proteins that now we are trying to bring into functional study to try to understand which one of these proteins can really have a causative role in the decline of organ function during aging. And so all of this data that I showed you up to now were obtained on intestinal crypts. So as I said, it's a very interesting sample because it provides information about not only the stem cell, but the environment where the stem cells are growing. But of course, this does not provide us whether the information, whether it is changing, on what kind of cell types are these changing occurring. And to do so, we need to go and be able to obtain protein profile from selected cell populations that we obtain from these tissues. And now I want to show you some preliminary data that shows the feasibility of obtaining data-independent acquisition-based protein profile also from pure population of FAC-sorted cells. And this is our strategy to obtain intestinal stem cell We are essentially taking advantage of a mouse model that was developed in Hans Kleber's lab, where intestinal stem cells are marked with a fluorescent reporter, GFP, under the control of the promoter of this protein, which is called LGR5, which is a cell surface protein which is expressed exclusively in the intestinal stem cells. Therefore, using this mouse, which has green intestinal stem cell, we can take the intestine, isolate the crypts, and use FACS to purify intestinal stem cells. And being very strict, in trying to obtain the purest population of intestinal stem cell, we can obtain around 50,000 of these cells from one animal, and that's what we then use to do our DIA mass spectrometry. We can, of course, this is highly reproducible, and this is what shows in this plot. So these are isolation 1 and isolation 2 indicates two different isolation of intestinal stem cells performed from two different animals in 2 different days. And here I'm just plotting the protein intensity in log transform for the quantified protein across the 2 isolation. And as you can see, we have very great reproducibility in aged matched and sex-matched animals. But what is the most interesting for us is if now we take 2 mouse genotypes that are known to have different intestinal pathologies, and we measure their proteome profiles, we can clearly distinguish between these genotypes at a coverage of over 4,000 proteins that we can cross-quantify across the sample. And this to us give us very good indication that we might be able to also find and identify the impact of aging also on these very special cell populations. A little bit of what we are heading. Of course, we are obtaining all these protein profiles, which are very interesting and very useful, but ultimately we want to understand which one of these alterations that we describe have a functional impact for the intestinal stem cell regenerative capacity and ultimately for organism homeostasis and lifespan. So, and for this we take advantage of 2 systems, one which is based is an in vitro regeneration system based on organoids. It was again developed in the Hans Kleber's lab, and these are essentially mini guts that one can grow in culture and engineer using CRISPR-Cas9 to deplete, for example, candidate proteins that come from our screen. And as far as organism homeostasis state is concerned, we are planning to use Drosophila as a model system, mainly because of its shorter lifespan, which is going to allow us to relatively quickly assess whether we can extend organism lifespan by manipulating proteins that we find to be affected in the intestinal epithelium. And just to give you a little bit of a teaser, using the data that I showed you, we already found 2 candidate proteins, which I'm showing here in this blue color, which are, when manipulated into our organoid cultures, are able to increase the regenerative capacity of intestinal stem cell almost 2-fold as compared to control untreated organoid. And this, of course, to us looks something which is very promising and encouraging to then move on to the Drosophila system and affect overall the impact of this protein on organism health span. And with this, I want to conclude and acknowledge all the people involved, in particular Nadia Geber, who is the PhD student working on this project in my lab, and all the other members of my lab, and of course the core facility Proteomics, which is headed by Jena, and that essentially form just one team with us. And of course our institute for funding support and biognosis, with whom we had a long-lasting exchange and collaboration in the last years, and they helped us a lot in setting up DIA in our lab. Okay, so I think I'm done with this, and Stefan, I will hand over back to you for questions, which we are happy to take.
Perfect, Alessandro and Joanna, thank you so much for this fantastic presentation and for the kind words about diagnosis. So we're looking forward to questions from the audience. As I said, you can ask them using the right-hand question function. Okay, while the audience is still typing, I have a question for you, Alessandro and Joanna. So what were the main drivers for you to explore DIA over other workflows? Maybe you can comment a little bit on how you honed in on which workflows were going to get you the types of results you were looking for.
I mean, from a technical perspective, we were evaluating, let's say the standard labeling, quantitative approaches, as well as, having got the Lumos in the lab, obviously the potential to do the DIA was very attractive, and we have done a lot of comparisons between the two workflows, and for us it's more, let's say, always the trade-off of what one can get out in terms of quantification and information within a certain time frame. And and also with studies like this where we have samples coming in over a very long period of time, PhD students don't want to wait until they've got their final sample to mix it with all their other samples, which one has to do in a standard labeling approach. And we in our comparisons, we've just found that actually, the depth of coverage one can get from this DIA approach is very similar to what one gets if one, multiplexes 10 samples and extensively fractionates them for the same amount of mass spec instrument time, which is, pretty much our rate limiting step in the lab. One can get really comparable depth of coverage of the proteome and quantitative information as well in terms of, missing values and all those kind of things. So, yeah, I would say that was our driver. And in this case, where the samples come in over a very long period of time, the DIA just makes loads more sense.
And another good point, I think, is that, particularly when we look at these purified cell populations, we are always facing, limited amount of material. And essentially, we realize that with DIA, we can get away with 2 injections, having built a good library. And for us, this is also an advantage because, it reduces the handling. You have just, to minimize the handling, which means minimize the sample loss. And we find it ideal also for this kind of sample. So the possibility of just prepping the sample, injecting it twice, and getting out as much data as possible.
Mm-hmm. Mm-hmm. Okay, fantastic. So a question coming in here, did you have the opportunity to check how the results overlap with RNA expression?
Unfortunately, not yet, but we are planning to do that, and particularly for the purified cell populations, but we have not done it yet.
Okay, fantastic. So a question actually directed at diagnosis, I see here. How do you help users of Spectronaut with the setup of DIA methods as Joanna showed? So if it's okay, Alessandro and Joanna, then I'll answer this one. So basically we provide on our website something called the Science Hub, which is a huge library of all the various app notes and manuals and methods and so on. So that's a great starting point if somebody wants to look into how to best set up methods. And then, of course, we have support at Diagnosis. You can actually see that email address to reach us here on the slide, and we provide you both on with advice on obviously the method setup on the LCMS side, but also on the bioinformatics side. So you can always reach out to us if you have questions around method setup, how to build optimal spectral library, etc. We have both written resources and are here for you to help you out. Okay, but back to Alessandro's and Joanna's work. So here's a question coming in. With your FACS sorted cells, about 50K cells, how much protein was analyzed? Did you also get about 4K proteins?
So yes, we got around 4K proteins, and we got out, I believe, in the range. These cells are also quite small, so I think we got around 3 microgram of protein from those 50,000 cells. and that gave us just about enough to perform 2 injections of the technical replicates.
Perfect. Okay, so let me see. Let's give the audience one more opportunity to ask further questions. It seems as if you guys really covered all bases so well already in your presentation.
That's very good.
Okay, so if there are no further questions from the audience, then I'd like to thank you again, Alessandro and Joanna, for this fantastic presentation. And we will, of course, share a recording with all the attendees. And if you have any questions, please don't hesitate to reach out to us at Diagnosis.
Thanks a lot for inviting us. It was great.
Thanks.
Bye, Alessandro. Bye, Joanna.
Ciao.